Great. So thank you for joining today and or whenever you're watching this recording. I'm Sharon Terry, the president and CEO of Genetic Alliance. And just a little bit by way of why I thought it was important to create Navigating the Ecosystem of Translational Science. And that is because, as most people know, I have two kids who were diagnosed in 1994 with a rare genetic condition. And I was pretty astounded at the sort of both cottage industry as well as highly regulated field of therapy and interventional development, including even device development and manufacturing, and the kind of morass it was, but the attempts that were made to keep it quite simple. And I appreciate those lovely chevron diagrams we've all seen that say, preclinical, phase 1, 2, 3, and post-marketing, as though it's a lovely chevron and we all go in a straight line. I also, in those early days, so the 1990s, I come in with a master's in theology at that point. I have no idea what it takes to do any of this or to find a therapy for the disease that suddenly we're aware of in my family, and then as I work with others and others' families. And I realized how much I didn't know. I also realized how much I was meeting advocacy leaders who thought that they should wait until a pharma company was knocking on their door to do anything. So very often in those days, and it's probably hard for some people to believe because so many advocacy groups get born now doing exactly this research. In the early days, in fact, I got thrown out of a major meeting because I stood up and said, we should be taking the bull by the horns. We should be taking the reins of this run amok wagon and making sure that we can drive the drug development. And actually, the leader of that major organization asked me to leave, saying, this is not our work. This is not what we should be doing. And so could you please leave the meeting? That was shocking to me, but didn't deter me because after all, the disease my kids had was worse than being thrown out of a meeting. So what I soon learned was lots of advocacy groups felt, no, not until that pharma company comes knocking on the door should I do anything. And what I thought was, and not because I was brilliant, but just because there seemed to be some logic in, if you wait to have a registry, a biobank, a mouse model, name it, until you have a pharma knocking on your door, then you're going to be 20 years behind being able to be working as quickly as you want to be. So that led to the invention of NETs, which again is, in some sense, I played with the word, saying we need a safety net, all of us, so that this is digestible by us, and we also need it to be meaningful and not be just a simple chevron diagram. So I'm going to share my screen. Let me just find the right window. Okay.
And while you're doing that, I will just put a shout out to Sharon because you're so understating the impact you've had in the early days and along the way. So thank you for that.
Oh, Dean, you've been a wonderful companion on the journey all these years. So, yeah. So what I did is I put together this map after a lot of conversation with some really terrific people. And I remember one of the first people I talked to was Francis Collins, who at the time was the director of the National Human Genome Research Institute prior to his becoming the director of NIH and now the advisor to Joe Biden, and said to him, I don't understand. And I would name a term and he would help me through it. And it became clear we really needed a more uniform way of trying to put something together. And so he and Chris Austin and Alan Guttmacher and me met like every, I don't know, 4 or 5 Saturdays at Genetic Alliance, brought some pastries, sat down and started to work out this map. And they didn't stick around kind of for the final putting it absolutely together. But we did go back and forth over it as I put it together. And the other piece that later on happened is Chris Austin decided many years later to actually take it and tear it apart with the drug forum at the National Academies of Medicine. And they actually wrote 2 papers, and I co-wrote them with them, so I co-authored them after this first paper that I wrote. And the map stood up to the test of their really going after it, and the 3 papers we've put in the resources so that you could have them. And they also created, the reason there was 2 more papers is one reviewed nets. They called it something else. I don't remember what it was, and it wasn't as catchy for me anyway. And then they also created one for biological entities, so that it wasn't just small molecule work. So my goals with this were essentially to create a more realistic model for the drug development process for neophytes, the kind of person I was, including basic researchers as well as advocacy leaders. Because the other piece I discovered was that while I felt like I didn't know what I was doing, I discovered that actually some of our basic scientists didn't know either, because they had never taken anything beyond the bench. And so that translational realm in those years was starting to get named T0, T1, T2, T3, T4. I was working a lot with folks at the CDC and so on, trying to understand how do we actually accelerate this translational science that in some sense was brand new in those years. However, it had been done, of course, years before as well. And so this helped basic scientists as well to say, not only do you figure out what an assay is on the bench, but you start to bring that forward into human development. To provide stakeholders with educational resources about each of the processes that feed into drug development, to highlight the on-ramps where stakeholders can become more involved. So that was also very important to me. How do we figure out where can I get on this morass and this non-linear, non-sequential process where I can make a difference, because not every one of us can make a difference in all of these places. To showcase stories of how others became more involved in drug development, and this webinar series will do some of that as well. And then to coalesce existing tools and resources for engaging in translational research. And I actually should say we should revise this 5th one to say existing tools and new tools. So as a result of this, for example, I began to work with Esperar, the drug repurposing nonprofit in Switzerland that I'm the president of, and Carolyn Kunt is the CEO of, and find ways for us to accelerate, in that case, repurposing. I became involved, obviously, with entities like Luna that you're going to hear about later in this webinar series, so that we would have robust registries. We work with a couple of different biobank vendors and have essentially morphed their tools so that they're accessible for us and very inexpensive. We created our own IRB and so on. So just to say, yes, we coalesced existing tools, and I'll show a few of those, as well as this webinar series will highlight new ones that were born, not necessarily as a result of NETS, but certainly as a result of our work in trying to say, where are the on-ramps? What tools do we need to make sense of this? And how are we going to use it? And then this just goes on to say, why was NETS created and who's it designed for? And I've said a pretty good piece of that. And how's it organized? So the NETS map is organized into 5 neighborhoods or communities of related processes, and they're basic science, therapeutic discovery, non-clinical research and development, clinical research, and regulatory requirements. And then we on our website presented 2 views of this NETS map, and you could either look at something like a neighborhood view. So if you click on the neighborhood view, you can see, and we color coded them essentially, that each of the 5 areas I just mentioned are available here in a neighborhood view, or you can go to the bigger complete view and all of the connections between the different pieces, again, to show how absolutely nonlinear this is and how interrelated and interdependent the various pieces are. One of the things I think that's important is that in each of these neighborhoods, and again, they're color-coded here for those who are colorblind, we have orange and green and pink and purple and blue. One of the things that was important for me in laying this out is to say there are interdependencies here, and not only that, but there are certain stakeholders who pretty much stay in their own neighborhood. They don't go to another neighborhood, but they have to interface with that other neighborhood. And so are we delivering the right thing? So for example, I think about a time when I was working with a lot of my scientists in the orange and in the green areas. So essentially looking at building cell lines and animal models, then also looking at what's the protein, what's happening here. and that they were not doing this in a standard way such that it could be then brought into the manufacturing regime, which is in this this blue area. So if they weren't doing the things that we needed to be able to industrialize with good manufacturing processes and lots of other standards that are required, then we were wasting time when they went from almost usually the academic environment into the industry environment. And so that became a very important piece for me, is how do we make sure that these little arrows, and sometimes many arrows, of interaction between the neighborhoods actually had some robustness to them, such that they were actually able to communicate with the next neighborhood or next piece. We also know that, as you can see by the lines, you might be in this area where you're starting to characterize the condition with some basic information and some basic bench models and so forth. But those are definitely feeding into and interacting with the clinical development piece, in the sense of being able to give the back to the bench some of the information that you need, and then the bench being able to bring forward into the people aspects that they're noticing, to see if we can see the same things in people and so forth. So anyway, that is that's the map. What we did for our work here, you can see as I start to roll over things, that we have definitions embedded in the map. So for example, clinical cohorts, I'm hovering over, pops up groups of individuals that share a common characteristic or disease. Cohorts can be helpful in organizing and recruiting eligible participants for a particular study or clinical trial. And then you can click on these and get even more information. And some of these are not filled out yet. We just actually moved the map over to a new place on our website and haven't brought over some of the data yet. But you can see that, in fact, there are some essentially resources available to individuals, and some of it is written in such a way that you can go deeper into, for example, therapeutic targets, relevant publications to those targets, related tools for those targets. And you can also imagine that this is a very big endeavor to keep up. And so Genetic Alliance has between 11 and 20 interns every summer, and some of them work on this map so that it does stay up to date, because lots and lots of tools are changing at an accelerated rate. I think the last thing I'll say here in this sort of more formal presentation of the map, and then we can go into any kind of discussion, is we understand that this is overwhelming, that there's a lot here, and that not every either advocacy group or even company will want to be involved in all these aspects. And again, we're very much looking for, how do we make this palatable? How do we make it easy to use? One of my dreams for it, should we ever get funding, and I even that is kind of on my shoulders. I haven't been able to spend as much time as I want focusing on the funding for this, but is that it actually have overlays on it, in the sense of, in the it's probably showing my age, but in the old days, a children's book might have just cellophane or plastic pieces that would add to the story. If you laid down more layers, there'd be more animals in the forest or whatever it was. I think that we could put overlays over this, and there could be some other information we could convey that way. The other thing that I've done a bit of is talk to people who do CAD, the computer aided design kind of work in engineering, because I think we could also make this like a Google map or Waze, the transportation GPS software, in the sense of, where are the bottlenecks? Like, where are a lot of people stuck? We intuitively know a bunch of those, and we actually know them also by evidence, but are there ones that we haven't sussed out yet that would be great to have some real-time data on? And how I foresee us doing that is, what if in a neighborhood, I would click on a neighborhood and find that, for example, Dean is in the neighborhood. Oh, and Dean is also working on an animal model. And Dean has run into a roadblock in such and such a place, and so have I. And so now, in a real-time interactive way, in this neighborhood, in this room, in this house, we could be talking to each other about that roadblock. And then we can leave some sort of mark on the map, the way I do with Waze, if I pass an accident or if there's a policeman on the side of the road that says there's a roadblock here. And maybe somebody else comes along and says, oh, no, I've figured that out. And we're able to have the kind of robust conversations we need about each and every one of these steps without making it overwhelming, without making it that, we'll have a week worth of webinar on the basic science, and then we'll have a week on the therapeutic discovery and non-clinical research and so on. I think being where you are with the problems you have today make it so much easier for us to be able to talk to each other about those problems and to find solutions. And then the very last thing I'll say is we will be presenting several other webinars that will highlight certain ones of these places within the neighborhoods and actually kind of blow them out into the very big topics they are. And those are our disease info search, which is data on 10,000 diseases and their subtypes that's entered by the individual advocacy organizations themselves. Has a whole lot of fields on research that we haven't revealed to the public yet because we're gearing up to actually finally launch another product called Gauge RX that, in fact, Esperer, Carolyn, and I worked on for years back in 2013 and 2014, whereby we were able to look at, so in these domains, then where are the sticking points for me as an organization or for my disease? So if I use pseudoxanthoma elasticum as an example, because I know that one very well, it's always been really difficult because we haven't been able to develop an assay. So assay development right here and a high throughput assay. No, we have an inside out vesicle of a bacteria that we have to reproduce in the lab all the time and can't do a big screen, a high throughput screen, the way NCATs can do for those of us who do have a high throughput assay. And that limitation has made our moving forward difficult. I also know that while we have a sense of what's happening in the disease, what the gene is doing and not doing and so forth, we don't have that as clear as I would like. And so that's another area that's a roadblock, a sticking point for us, and one that we continue to work on. And then the other one is that we don't have biomarkers. So as we're in this pink area of going through clinical trials, which we are right now, we don't have a good biomarker. We don't have a good endpoint, which is often the case for rare conditions. And in our case, particularly one that moves as slowly as it does. It's fabulous that it's slow. It's terrible that it takes 20 years to see any change. And so we don't have as good outcome measures and therapeutic clinical endpoints as we need. And so those are places also that that we can work. And so those kinds of things we've now put into an algorithm such that it spits out a score. And we have a group of students from the University of Michigan, 8 of them in information science, that are working on this fall and winter. To actually produce a tool then that we can look at any disease or any advocacy group and see where these places where there are roadblocks, where there's more need, and where we score low. And not to score low because it's a terrible thing, but to score low to say, here's a place we really need to do some work. That brings us to several of the other webinars after that, which will be, we will be having a presentation by Luna, which runs our registry and our what we call community-driven innovation, which susses out what's important to people, because keeping them engaged this whole process is very important. And then Esperer, who will be bringing forward not only their own expertise in things like trial design and clinical endpoints, and repurposing drugs, working with pharmaceutical companies in such a way that novel and ethical pieces can be embedded, as well as Esperer will also be putting forward an RFP to award an advocacy organization resources to move a molecule, an asset, through repurposing into the market. And so that's a phenomenal offering that will happen. And this webinar series builds toward that. And then the idea, of course, that all of these things need to be scalable, that we don't want any of this to be one-offs, and that was always my goal in making these sorts of maps, is that we actually look at how it is that we can bring these things forward more quickly, more inexpensively, and so forth. So I'm going to stop there. That was plenty of information, I think, and open the floor to questions here. And then we're happy to have questions if you're watching this at a later date. My email is quite simple, Sharon at geneticalliance.org. And we even have ways on the website for you just to click links to give us feedback or ask questions. So the floor is open.
Well, I have an observation, which may take us a slightly different direction or create a compendium map. And Sharon, I think you're right that a lot of people will see this on the video, and perhaps new organizations, and they say, "Oh my goodness, I don't even know where to start." Right? This is overwhelming, and indeed it is. It is not a process that gets done in just a few weeks or a few months. But a couple of observations that are, I think, of particular value to advocacy organizations, because we do partner most often. Some organizations take this all on themselves, but more often I think they partner with pharmas or big pharmas or new companies or institutions that are doing this research, where there is some momentum and some experience, and perhaps some ruts they're stuck in as well. But places that advocacy can be particularly impactful, particularly as you're trying to figure out what you do, is the upper left. At least I think this is where you might place it, Sharon, the data repository. So what kind of data? How can we build up data and information about our organizations and our communities? And down in the lower left, that yellow box, which you've labeled post-marketing, but I think that a lot of therapies these days, and we're running into this with MLD, gene therapies is kind of the strategy that's got the most momentum right now. And these issues of having good, and I saw clinical practice guidelines, I think, in another box as well, maybe in the pink box, but starting to develop what are the standards of care and the practice guidelines and the values or the burdens of the disease, so that you can justify the reimbursements and so on. So there are places that we can come alongside existing projects or start to do work in advance of launching new projects, so that whoever it is we end up partnering with, we bring valuable things to the table. It's not just knowing the name and the email of patients to recruit for clinical trials like it used to be. It's so much more. We can be so much more impactful and a much better research. And as you would say, Sharon, a peer at the table. We're not just looking in through the outside window. We're sitting at the table through this process.
Yeah, terrific, Dean. Thanks. Yeah. And even the last couple of months I've been saying, and not only appear at the table, but decide where the table will be, what it will look like, who else gets invited, what's the menu. Yes.
Right,
yeah.
right. And doing the dishes.
Yeah, that's right. Yes, we definitely have that part as well. Great. That's very helpful. Thank you. You're right that I think in a lot of these things, so we certainly run into this with helping groups get onto registries, which we've done now for many hundreds of groups. People feel like, oh, it's overwhelming. I got to spend a couple of years planning. And there's that saying that says the best time was 20 years ago. The second best time is today. And I firmly believe that in this work. And so in all of these things, we're encouraging people to figure out, yes, where is it that I can start to work, and then where can I get help to do more of the work that is required here, so that I'm actually accelerating this pathway. With 7,000 rare diseases and only 300 and something therapies, we know that there are many, many more neglected diseases than there are scientists for. And I think part of our desire in EngageRx too is, how do we make it more obvious that a lot of us are really ready for this kind of work, for this kind of trials. And it'll be part of what we'll see in the Esperaire webinar, as well as, and the many, many molecules that are sitting on shelves in pharma companies that they don't mind repurposing, but we need some basic building blocks in place for it to make it worthwhile for them to do that.
Yeah, and Sharon, thank you very much for this great presentation. And to add to what you said, something that could be very interesting and that we could think about when you talked about layering on this net map, some, let's say, examples of success and also failures, and to show basically on case studies the importance of all these blocks coming together to be able to ambition to bring a treatment to the patients, but also to show that in the type of disease we are in, there is a bit of flexibility and creativity in the way we can think of advancing these therapeutic opportunities. And that should also be a strong message to the community in terms of the willingness of regulators, as long as we don't compromise quality and safety of the patients, and we are able to show impact for the lives of these patients. There is some flexibility to be brought, and I think a great value would be to have, as I said, concrete examples of success and also failures that would also potentially help the community thinking also what does it mean for the disease and the community I'm caring for.
Yeah, thanks, Carolyn. In fact, when I was looking here at the different different blocks that we do have some information on, and I was thinking we did build in this section testimonials, how we did it, and wanted advocacy organization to come forward and put these in. And there are some that just aren't deposited here yet, but that's a great point. And I think what we'll do is we'll make a foray, a pitch to get some of these case studies, testimonials in so that we can layer those in as well. That's great.
And Sharon, you can count on us to offer in the disease we are working in to also think about how we could structure these case studies, because I think it would really also put a context, a live context to this great map that should really help us think through the way we should think about drug development.
Yeah, super. Yeah, and we have published 3 case studies from our work with Luna that definitely could be deposited in here as well, because they illustrate kind of the gamut as well. Super. Okay. Well, this is all that I hope to achieve with this first webinar, is essentially to lay down this massive bedrock. There's lots, as I said, that can be done with it. I think there's a lot of places for this kind of interactivity, both case studies as well as understanding the roadblocks and the failures as well. And then what we're going to do through this series is simply build on this so that, to Dean's point, advocacy groups can see their on-ramp and don't have to feel like they have to bite off this whole thing. There is this tension in this world about the chevron diagram is beautiful because it's simple. On the other hand, this is really complicated and could seem overwhelming, and that there's somewhere in between that all of us will feel more or less comfortable, and that'll be a place for us to give some time and energy. Okay, well, thank you guys. And again, we're open to comments and questions and suggestions, and it's a very interactive process. You can see that there's a lot of space for us to be depositing more information here, and we're happy to add it as you come across anything that's juicy and good and useful to our communities.
Wonderful.
All right. Have a good evening, morning, afternoon in the different time zones.
Thank you very much, Sharon.
Thank you. Bye-bye.
Bye-bye.